Hello and welcome to the B3.3 Muscle and Motility Review video. This is an HL only, so make sure if you're an SL you don't need to watch this. Now, Standard 1 says that movement is a universal feature of life. So all living things have adaptations that allow them to move. What does that actually mean for them, though, depends on the species. And so they have adaptations for movement. Some are very obvious. So we have our motile species. These are like us. We could do cheetah, right? So cheetah has its special legs for running really fast. It's a good example because you need to know it in another unit. And then we have our sessile species. Our sessile species are very different from our motile ones. For sessile species, that means they do not move on their own. And so they require something else to help them move. So for example, for our plants, right, they have their seed dispersal. So they have adaptations to disperse their seeds, but they're not going to move them themselves, right? So they're going to use either an animal or the wind or water. They have adaptations to help move, but they're not doing it themselves. So they do not move on their own. But they still have adaptations to move. Now, Standard 2, we're just diving right in. Standard 2 is the sliding filament model of muscle contraction. Now, what you have is, you have this thing called a sarcomere. The sarcomere is the contractile unit. This is the part of the muscle that does the contracting. And it's made up of a couple of different parts. So your sarcomere is composed of these things that look like this. You have the thin filament, or actin, and you have here in the middle you have myosin, or the thick filament. And here at the end, you have titin. Okay, and then you would have that on the other side as well. Oops. You have the other sides of actin, like this. You also have it on this side as well, too. Actin, actin. Okay, so myosin goes through the middle, and then you have actin on either side. And this area here can shrink or grow. And so that's called the H zone, and that can shrink or grow. And so for the sliding filament theory, or model, what you need to know is that basically it's all about myosin binding to actin. Myosin binding to actin. The myosin does not move, but it's going to pull the actin. Okay, so here we go. So step 1 is the myosin heads will bind to actin and release ADP. Alright, and so when it's doing that, it looks like this. The myosin head is fully extended, and it's binding to the actin, and so you're releasing ADP. So the ADP is coming out. Now, that's step 1. Step 2, myosin head will contract and pull actin. And so it'll still stay the same, but then it's gonna go from this angle to this angle. And so it's gonna move to this position. And so then you can see, if I got it like this, the actin has now moved that way. Okay, so it's like you grabbed on and you pulled like that, and so you pulled the actin that way. And so this is actin, this is myosin. Okay, so it's changed conformation, the shape, the way it's pointing. Then from there, what's going to happen is the ATP will bind to myosin head and cause it to release actin. So then it'll look like this. Okay. From there, the ATP will dephosphorylate and release it. So it'll go like this. You still have your actin, you still have your myosin, and it now is going to go back to this position, and it's going to have ADP attached. So you release a phosphate and change position of myosin head. Then you're going to go back to step 1. So you release ADP and bind to actin. And then it'll just keep cycling through that. So okay, so you're going to go back to this. Oh, we release the ADP and it binds. ADP is released. And you can see how then it will pull again, and so it's pulling, it'll do the whole thing again and then pull the actin when it repeats. So that's the sliding filament contraction. Now we said at the beginning that in the sarcomere there's titin. And so titin is like a spring. It's like a spring. And so it is that part at the end of the myosin, and it prevents overstretching because it is a pretty strong protein with a lot of elasticity. And so it prevents overstretching of the muscle, and it recoils and helps relax muscles. So it helps relax muscles. This allows it to work together with antagonistic muscles to relax, because you can only have power when contracting. So when you're relaxing, there is no power to it, and so titin is the thing that helps it recoil, as well as the antagonistic muscles. Now, this one's kind of interesting because we're kind of going backwards, but I'm trying to follow with the standards. We have here a motor neuron and your muscle. And so this is the muscle fiber. We have here the motor neuron. And we have a neuromuscular junction. And so how this works is it will do the same thing it normally does. Neurons will do what they normally do, and it'll release a neurotransmitter. In this case, it's acetylcholine. The acetylcholine will then bind to a receptor. When it binds to the receptor, it will open calcium channels and cause the release of calcium ions from the sarcoplasmic reticulum. The sarcoplasmic reticulum is just a specialized version that holds calcium ions. So it causes that to open, and then those calcium ions start muscle contraction. This was in the old standards. I am not 100% sure you need to know it for the new standards, but I'm going to say it anyway. On actin, that thin filament, there's this thing called tropomyosin. And tropomyosin blocks the myosin binding sites. And it has to be moved before you can start muscle contraction. And it's moved because on it there are these things called troponin, and troponin binds calcium ions. And when it binds calcium, it causes a shift in tropomyosin, which enables the sliding filament model. So when I said we were going backwards, that's what I meant. So we didn't talk about, well, why can sliding filament start? And so that's why it can start. So it starts with the motor neuron, releases acetylcholine. That acetylcholine hits a receptor, which causes the release of calcium ions from the sarcoplasmic reticulum. And then the calcium ions will bind to troponin, causing tropomyosin to shift. And so like underneath here, these would be like the myosin binding sites. And so when it shifts, then it exposes those so that you can do the sliding filament model that we learned about in Standard 2. All right, now the good news is those are the hard standards for the muscle unit. From here, it gets a little easier. Standard 5 says that skeletons are used for anchorage of muscles and as levers. So when we talk about levers, we're thinking about like a screwdriver or a drill. Like the larger you make it, the more leverage you have. And so if you've ever tried to use a wrench, the longer the handle on the wrench, the more force you can apply because of how torque works. It's the same with your bones. The larger they are, the more leverage you can get, but then they get heavier, right? And so there's trade-offs, just like there is all throughout biology. But these are the two main things that it works about. Now, there's a little thing though. We have endoskeletons for vertebrates. That means it's inside, and the muscles are on the outside. And then exoskeletons in arthropods. So it's on arthropods, and so these are on the outside. And so what you've got going on here, right, is muscles are on the outside versus muscles on the inside. So they function very similarly, but where the muscles are attached is different, right? So in a very, very, very oversimplified insect, you have to attach the muscles on the inside. But on humans, right, you have the bones and the muscles attached on the outsides like that. So this would be exoskeleton, and this would be endoskeleton. Right, so the attachment points are very different. All right, then we need to know our synovial joints. Synovial joints. Specifically, you are expected to know the synovial joint that is your hip joint. You're expected to know the hip joint. And so you have the pelvis, and then your femur, wow, that's actually like really good. your femur sits in it like this. And so your synovial joint is right here. This is the actual joint, is this part right here. Okay. And so then you have inside there, along the edges of the bones, you have cartilage. So lining the edge of the bones, you have cartilage. And this is for cushioning and for flexibility. Inside here, you have synovial fluid. So synovial fluid is your lubricant and also for cushioning. And then you have some ligaments. Ligaments hold bone to bone. And then you have your muscles, right? So your muscles would be like over it. And so like that'd be like your quad. You don't need to know the name of it. And then your muscle, remember, so tendon will hold muscle to bone. So you have tendons that are holding the muscle to the bone. So your quadricep muscle would be held in place by a tendon. And so they're free moving and they're enclosed in a capsule. And so right near where the ligament is, there's also the capsule. And so there's capsule that holds all that fluid in place. So those are your synovial joints. All right. Standard 7 says you should be able to measure the range of motion of joints. And so you use a tool that looks something like this. So this is stuck in spot, and then you have another one that can rotate. So this one is free to rotate about this one that's stuck, and you use that. So you hold that at the joint, and then you measure the flexibility. So you, like, this is along one bone, and then the other one follows the other bone to test the range of motion. You can also use computer simulators to do this. This device here is called a goniometer. Okay. That is standard 7. Now, standard 8 is all about antagonistic muscles. So these are muscles that work in pairs. So when one contracts, the other relaxes. You have a lot of them, like your bicep and tricep. Pretty much all of your muscles are antagonistic. The main ones we need to know for the IB standards, though, we've already learned about. Those are your external intercostal muscles. Whoops, I wrote internal. Internal intercostal muscles and your external intercostal muscles. And these are found along your ribs. And these are all about moving your lungs. So the internal intercostal muscles move your lungs inward, and your external ones cause your lungs to expand. So they pull them outward. And so when this one is contracting, then this one must be relaxing, and vice versa. So if you put your hand on your chest, you can go, and you can feel it moving in and out. So they're moving in opposite directions. Titan, right, has a role in this, right? It stores the potential energy in that spring-like function. So it stores potential energy. So when it's relaxed, Titan is storing the potential energy for you. So they're moving the rib cage in opposite directions. And that is standard 8. Standard 9 says, why do we move? And they call it locomote. What a great word. Locomotion, or movement. There were 4 main things we need to know. Foraging for food. So when you get out of your bed and you go to the kitchen. Standard 2, escaping from danger. When you're getting yelled at by a parent and you decide to run to your room. Or, the classic example, you're being chased by a polar bear, which also activates your fight for flight. Look at those cross-cutting concepts. 3, searching for a mate. that'd be like Endler's guppies, a real example. And then migration. And so if you are in HO, which obviously you are if you're watching this video, migration, right? We need to develop the reindeer is one of the standards in the Arctic mouse-eared chickweed, as well as your great tit in the caterpillars in Europe. Right, and so those are our reasons for locomotion. Pretty simple. I like that standard a lot. Standard 10 is all about our dolphins. Oh yeah. Or marine mammals. So they have a fluke that moves up and down to power them through the ocean. They have flippers instead of like a hand or like feet to make them swim through the ocean. They're streamlined to reduce water resistance. And they have special lungs and breathing that allows for periodic breathing, so they can take large breaths, hold it, and close their airways. Close their airways. This enables them to stay under for longer periods of time and to not let water into their lungs. So, right, marine mammals are animals that used to live on land but now live in the water again. And so they have lungs like land animals, but you obviously don't want water in there. So they have the ability to close their airways. And so often they have like a blowhole, like if we're talking about dolphins, they have like a blowhole that allows them to close it off. But your seals also have the ability to close off those airways too, such that they can hold their breaths for longer. And this is the end of B3.3.